Hello friends. Well, today in this video we will talk about DNA, the introductions part and all these structure of DNA. Well, when we say nucleic acid, we say that nucleic acids are made up of heterocyclic bases, purines, pyrimides, sugar, maybe 2-deoxyribose or ribose, and phosphates. And you say that nucleic acids are monomer of nucleotide. We have talked in detail about nucleoside, nucleotide, phosphodiester, and these nucleotides are attached with phosphodiester linkage, and how this nucleotide sequence can be demonstrated. We have talked all these in detail in our previous video. Well, when we say classification of nucleic acid, the nucleic acids are classified on the basis of the nature of the sugar moiety present in the nucleic acid. So the nucleic acid has been classified into two types: ribonucleic acid or RNA and deoxyribonucleic acid or DNA. As the name suggests, you see here, if you have this sugar moiety, that is ribose, okay, you say that the nucleic acid is RNA. Similarly, if you have this sugar unit, that is 2-deoxyribose, the nucleic acid is DNA, that is deoxyribonucleic acid, what we are discussing today. Now, when we talk DNA, we say that DNA is present in all living cells except plant viruses. We also know that DNA are hereditary materials. That is, when we say hereditary material, you are talking the material, DNA is responsible for transformation of all the characteristic of parents, from parents to the DNA are responsible for transfer of characteristic of parents from parents to their offspring. So you say that DNA are hereditary material. Now also various cell activities information. Now you see here, information of different cell activities and life process are encoded in DNA molecules. Okay. Now let us talk the structure of DNA. Well, when we talk structure of DNA, we discuss the structure of DNA in two heads. That is primary structure of DNA strand. Primary structure of DNA strand. All this we will be gradually knowing, that a strand and this double helix and all this. And the second head is secondary structure of DNA. Secondary structure of DNA is also called Watson and Crick model of DNA because they discovered it. It is also called nowadays B-DNA or B conformation of DNA. So all this, that is Watson and Crick model of DNA, B-DNA, B conformation of DNA, and secondary structure of one thing. Well, when we say primary structure of DNA strand, obviously DNA strand means you are talking one strand, that is DNA, okay, DNA strand. But how it is made up of? You know, when we talk DNA strand, obviously it will be composed of, as you know, that it will be composed of 2-deoxyribose, the structure is there. The sugar moiety is 2-deoxyribose in DNA, you know it. Then you have adenine and guanine, the purine bases, and you have thymine and cytosine as the pyrimidine bases. Okay. Now you also know that DNA is a polymer. Repeatedly we are saying that it is polymer. DNA is a polymer. Polymer of what? What is the monomeric unit? Monomeric unit are deoxyribonucleotides. Deoxyribonucleotide. You know that nucleotide, that is made up of heterocyclic bases, sugar and phosphate. The phosphate is at 5' position. And what are these deoxyribonucleotides? Now you see here, these are deoxyadenosine monophosphate. Okay, you also called it deoxyadenylate. Then you have deoxyguanosine monophosphate, that is deoxyguanylate. Similarly, you have deoxycytidine monophosphate, you also call it deoxycytidylate, and deoxythymidine monophosphate or thymidine monophosphate, that is deoxythymidylate. Now here you can see that you are writing either DTMP or TMP. Since only thymine is present in DNA, so if you write TMP, it means you are talking DNA. Okay. Now these ribonucleotides, you see here, the monomeric deoxyribonucleotides are attached with 3' 5' phosphodiester linkage bonds. Now when you say 3' 5' phosphodiester linkage bonds, is the connecting the two monomeric deoxyribonucleotide. The experimental verification comes from enzymatic hydrolysis. The presence of 3' 5' phosphodiester linkage in deoxyribonucleotide strand comes from enzymatic hydrolysis. When DNA is subjected, you see here, when DNA is subjected to pancreatic hydrolysis with pancreatic deoxyribonuclease, what you get? You get a mixture of oligonucleotide. When you say oligonucleotide, obviously you are talking nucleotide having more than 2 nucleotides. So you get a 4 nucleotide average. Most of the time you get 4 nucleotide oligonucleotide. That is, average is number of nucleotide in the oligonucleotide obtained by hydrolysis is 4. And what it is interesting, that at 5' end of this oligonucleotide you have 5' phosphate residue, while at the 3' end the 3' OH is free. Okay, and when this oligonucleotide is subjected to hydrolysis with another enzyme, that is spleen phosphodiesterase, okay, it is also called deoxyribonuclease 2 enzyme. Okay, what you get mixture of deoxyribonucleosides. So what is the inference? The inference of this particular enzymatic hydrolysis of DNA, since what you are getting a 5' residue at the 5' end, and 3' you are getting at the 3' end, OH free. So what you have, this experiment inference or confirms that the nucleotides are joined together by 3', 5' phosphodiester linkage, and the structure of the DNA strand, the structure of the polymeric DNA strand formed by the monomeric deoxyribonucleotide is linear. Okay.